deep research for an EEG filtering. We got this website. So we have the interactive report. No, wait. Recording engineering precision and clinical expertise in EEG interpretation. Okay, it's having all the sources. Yeah, well, the point of it is that we can actually turn it into a web page. So we have the mirage, the signal chain, patient electrode amplifier, ADC, it's analog to digital conversion, software, and the interpreter, which in most cases is a clinician. We don't have a clinician, so we're using AI just because it's too expensive to have access to a clinician. So we have cognitive biases. Yeah, it's really nice, really nice. Confirmation bias, anchoring bias, availability, heuristic, premature closure, accepting a diagnosis before it has been fully verified, ending inquiry too early. Yeah. Can only imagine how often does that happen. And yeah, we don't want to just talk about the problems. We have some solutions. So education, better standards, intelligent systems. So we'll be focusing on those for obvious reasons. Well, we are not in academia. We are not setting standards, but we can design simulations and train models. So there you go. So that's a nice little thing from those reports. We have the report. We can download it as PDF. Obviously, we had things like that on the website already. Go check it out. You can create a web page, infographic, a quiz. A quiz will be nice. You can actually learn, test your comprehension. Ooh, something. I thought something happened to Gemini. I thought we lost our CEO. No, we still have it. Still have a CEO. Yeah, just generating like infographics and content out of that very long report, which obviously no one will read, but yes, we can make it available as PDF. Yeah, we can do a quiz, audio summary out of it. Yeah. So yeah, this video brought to you by Gemini. Don't remember what the prompt was. Yeah, notebook LM is a bit interesting that way. Oh yeah, well because there's so many different ways of using it as well. Yeah, and also ask for 8 to 10 minute summary, but give me 15. So yeah, not sure. I'm not even sure if it's following prompts at all. I suspect it's not, but it is. It seemed to be true to the sources added in the conversation. So yeah, so we have a bunch. We'll do a summary. Go check the website. Let us know what you think. Might load this brain because I might start losing frames as I do. So I load this tool. Do I lose frames? No, I didn't drop any frames. Didn't drop a single frame.
Imagine trying to listen to the most intricate symphony, but you're stuck in a really crowded concert hall.
Right.
There's coughing, people shuffling, maybe a phone goes off. That's kind of what it's like trying to decipher the brain's electrical activity, isn't it?
That's a great analogy.
Yeah.
Yeah, it's definitely less serene string quartet, more like a full orchestra competing with a pretty unpredictable audience.
Totally. So today we're doing a deep dive into electroencephalography, EE for short.
Yeah.
Our mission, to shortcut your way to getting a handle on how these tiny brain signals are captured, what kind of noise messes them up, and believe me, some of that noise is weirdly personal, almost darkly funny.
Mm-hmm.
We'll look at the clever ways researchers try to clean it all up, and then the real kicker, why for some really advanced stuff that noise might actually be helpful.
Exactly. We've got a good stack of sources lined up to walk us through this whole intricate process.
Okay, let's unpack this then. We know EEG measures brain activity, but how precisely do we even pick up those super faint electrical signals from someone's head?
So at its core, EEG is a non-invasive way to measure the collective electrical buzz of huge groups of neurons firing together synchronously.
Okay.
Think of it like trying to hear the orchestra's overall sound from just outside the concert hall door.
Yeah.
You're not disturbing the players inside.
And how do they physically capture that sound? You can't just stick electrodes anywhere, I assume.
Oh, definitely not. It's all about precision. The gold standard is something called the 10-20 system.
Okay, 10-20.
Yeah. Imagine drawing really precise lines on your scalp. You use specific landmarks, like that little dip at the top of your nose.
Right between the eyes.
Exactly. And the inion, that bony lump at the back of your head. From those points, you place 21 electrodes symmetrically. They're measured at specific 10% and 20% intervals of the skull's perimeters.
Hence the 10-20.
Precisely. And this standardization is absolutely crucial. It means recordings from different people, different labs, different times, they're actually comparable. That's vital for research, for diagnostics.
Okay, so we're all hooked up nice and neat. What are we actually hearing? Does the brain play different tunes depending on what it's doing?
That's a really good way to think about it, yeah. EEG doesn't just measure activity in general. It reviews these distinct electrical rhythms or waves that tell us what the brain is up to, both in time and across different areas.
Like different musical scores?
Sort of. For instance, if you're relaxed, maybe eyes closed, chilling out, you're likely producing rhythmic alpha waves. They're typically between 8 and 13 hertz.
So meditation mode equals alpha tune.
Pretty much. Now, if you're really focused, concentrating hard, or maybe even feeling a bit anxious, we see faster beta waves. Those are anywhere from 14 up to 50 hertz.
Okay, faster for more active thinking.
Right, and then there are slower ones too. Theta waves, that's 4 to 7 hertz, often linked to mental stress, maybe drowsiness, and even slower delta waves, 3 to 5 hertz, which really dominate when you're in deep sleep.
Makes sense. Deep sleep, slowest rhythm.
Exactly. And then way up at the fast end, you've got gamma waves. We're talking 40 to 140 hertz.
Wow.
Yeah, those are associated with peak focus, intense thinking, maybe even consciousness itself. It's this incredibly complex mix, millions of neurons firing, synaptic currents, even the physical shape of your head plays a role.
But the brain isn't the only thing making electrical noise up there, is it? And this is where it gets, well, kind of funny in a dark way. The noise can be very personal.
Oh, absolutely.
Yeah.
An artifact in EEG is just any electrical signal that doesn't come from the brain itself.
Okay.
It's all that extra chatter in our concert hall. We usually split them into two main types. Physiological, coming from your body, and non-physiological, from outside sources.
Physiological. So interference literally generated by us, our own biology getting in the way.
You got it.
Yeah.
Your normal bodily functions can create huge interference. Think about simple things. Moving your eyes, blinking.
Just blinking, really?
Oh, yeah. Even chewing or grinding your teeth, that's called bruxism, or just swallowing. These muscle contractions, they generate electrical signals way, way stronger than the brain's own signals.
No way.
Seriously. One source we looked at showed how a single swallow lights up almost all the electrodes with these huge spikes.
So your brain's trying to compose this masterpiece, and your jaw deciding to chew is like a sudden, deafening drum solo.
Small chuckle. That's pretty much it. And it's not just muscles. Your heartbeat, the EKG signal, even just breathing, sweat glands.
Sweat glands.
Yep, skin potential changes. Or if someone has an artificial pacemaker, that's another source of electrical noise. It really is kind of darkly humorous that just being alive makes it harder to listen to your brain.
Especially someone's stressed, right? They probably move more.
Exactly. Anxiety, stress, finding it hard to stay perfectly still, that often makes these physiological artifacts worse.
And then there's everything else, the whole world trying to butt in.
Precisely. That's the non-physiological stuff.
Yeah.
It can be mundane things like a bad connection where the electrode meets the scalp, maybe it's placed wrong, or faulty wiring.
Technical glitches.
Right. But it also includes electromagnetic fields just floating around in the environment. The hum from the power grid, 50 hertz here, 60 hertz in the US.
The mains hum.
Yeah. Other medical equipment, if you're in a hospital, your cell phone, even fluorescent lights, all that can interfere. Sometimes you see what's called an electrode pop.
A pop.
Yeah, it's like this abrupt, spiky jump in the signal when an electrode just momentarily loses good contact. It really underscores there's a constant battle researchers fight just to get a clean recording.
Okay, so with all this, unwanted input, crashing the party, the huge challenges, how do you filter out all these uninvited guests without accidentally throwing away the important brain signals you actually want?
That is the million-dollar question. It's this incredibly delicate balancing act. Whatever cleaning method you use, it's crucial. It doesn't create new problems, new artifacts, or just erase valuable neurological data.
So what are some of the, let's say, classic ways people have tried to tidy up this messy signal?
Well, historically, the methods range from pretty simple to really quite complex. If you're looking just at the signal over time, you might do basic amplitude thresholding.
Meaning?
Like if the signal suddenly jumps to 6 times its recent average height within, say, 10 seconds, you flag it.
Yeah.
Probably an artifact, right? Too big, too sudden.
Okay, like a that doesn't look right filter.
Kind of. Or you can get more subtle using mathematical models. There's one called a quadratic linear parabolic model. Its parameters, how they change, can help distinguish the shape of a normal resting signal from, say, a muscle twitch artifact.
So looking at the shape of the wave, not just how tall it is.
Exactly. Then moving into the frequency domain, looking at the different rhythms, there are techniques like musicus, multiple signal classification.
Music. Okay.
Yeah, it cleverly works by estimating the frequency profile of the noise itself, so you can then subtract it out. It's pretty effective, but it takes a lot of computing power.
Resource intensive.
Very. Another approach is the empirical wavelet transform. This one's cool. It adapts its filters to the signal itself, almost treating the EEG data like an image, finding patterns.
Turning brainwaves into pictures again? That's neat. A smart way to zero in.
It really is. And beyond just time or frequency, statistical modeling is huge too. Like you can try fitting the EEG data to a specific statistical distribution, a T-location scale distribution, for example.
How does that help?
Well, the specific parameters of that fitted distribution, its shape, its spread, they can act like a fingerprint. Different artifacts like teeth grinding or a cell phone buzz might distort the distribution in characteristic ways compared to normal brain activity.
Okay, so using stats to spot the odd ones out. Clever.
So we're trying all these ways to pick out the brain's true voice from all the surrounding noise. And I think you mentioned some of the really heavy lifting involves multivariate analysis. What's going on there?
Yeah, what's that about? Sounds complex.
It means we stop looking at each electrode signal in isolation. Instead, we look at the relationships between all of them, or many of them, at the same time. Consider the whole set of measurements together.
Okay.
A really popular approach here is blind source separation, or BSS. The basic idea is to try and unmix the recorded signals, assuming they came from different independent sources that all got jumbled together at the electrodes.
Like trying to separate out individual instruments from a bad recording where everything got mixed onto one track.
That's a perfect analogy. Principal component analysis, PCA, is a classic BSS method. It finds the main directions of variation in the data, trying to boil it down to the most important underlying patterns.
Sounds useful.
It can be, but honestly, it's not a magic bullet. PCA often struggles to cleanly separate things like eye movements or muscle activity or even the heartbeat signal, especially if those signals are roughly as strong as the brain signals you're after.
Right.
That's like trying to untangle spaghetti. But some strands look almost identical to others, and they're all kind of sticky.
Frustrating. That almost but not quite feeling.
It really can be, which naturally leads to the question, if PCA isn't quite cutting it, what's next? And that's often independent component analysis, or ICA.
ICA. How's that different?
Well, ICA takes a crucial extra step. It specifically tries to transform the data not just into uncorrelated components like PCA, but into statistically independent components.
Independent.
Meaning the sources are assumed to be generated by distinct processes that don't influence each other. Think of eye blinks and alpha waves. They come from different biological generators. Yeah. ICA is designed to exploit that independence to unmix them more effectively.
Oh, okay. It leverages the idea that these signals should be separate.
Exactly. And many studies show it works quite well for removing common artifacts. Interestingly, there's a little trick. Applying a high-pass filter before running ICA can sometimes boost its performance for certain analyses.
That pre-filter helps.
Sometimes. But there's always a trade-off, right? Doing that can negatively affect something called dipolarity.
Dipolarity?
Yeah, it's basically a measure of how well the separated component resembles a signal coming from a plausible single-point source within the brain. So you might get cleaner separation with the filter, but the resulting brain components might look less biologically realistic. It's that classic engineering compromise.
Fascinating. Okay, so that brings us towards the cutting edge. How are the latest computational tricks, AI, machine learning, tackling this never-ending cleanup job?
Yeah, we're seeing some really exciting new things emerge. For instance, there's a new metric called oscillation energy, or E sub alpha.
Alpha.
Right. Its goal is to characterize genuine brain oscillations much more precisely. See, traditional methods often look at broad frequency bands, right? Like 813 hertz for alpha. But that often lumps together the real oscillation peak with just general background noise or non-rhythmic activity in that band. EEG tries to specifically amplify and separate out the peak of the oscillation from that background mush.
So it's like really fine-tuning the receiver to get just the core melody, not the static around it.
That's a great way to put it. It makes it much more sensitive to subtle changes in brain activity and less swayed by just general background shifts.
That sounds like a big step for understanding specific brain functions.
It potentially is. And then, of course, there's the huge wave of artificial intelligence and deep learning. These tools are just fundamentally changing how we analyze EEG. We're moving way beyond staring at squiggly lines on a screen.
How so?
Well, for example, people are tuning large language models, like the ones behind chatbots, but feeding them vast amounts of neuroscience knowledge.
Wow.
Yeah, to help with diagnosing diseases from EEG data, even generating explanations that sound like a human expert's reasoning. It's pushing the boundaries, turning that raw signal into potentially actionable clinical insights.
Here it comes, right? The kicker, the aha
Yeah.
moment that flips things around, especially for deep learning. It's got this wonderfully ironic twist.
It really does. Okay, so when you're preparing EEG data to feed into these powerful deep learning models, the standard thinking, the conventional wisdom has always been more preprocessing is better.
Clean it up as much as possible.
Exactly. Cleaner data, purer brain signal. Presumably the AI will learn better, perform better. Makes intuitive sense.
Yeah, totally. Garbage in, garbage out kind of thing.
But recent research, some really critical studies, have found something genuinely surprising. First off, feeding totally raw EEG data, like straight off the scalp with zero cleaning, that generally leads to pretty poor results.
Okay, so raw's bad. That fits.
It consistently ranks last in accuracy tests.
Yeah.
But here's the turn. It turns out that often minimal filtering, maybe just basic stuff like removing the mains hum, often hits the sweet spot. It gives the best balance between how much work you put into cleaning and how accurate the model ends up being.
So not too much, not too little, the Goldilocks zone of data cleaning. But wait, there's more, isn't there? The ironic part.
This is it. The really sophisticated preprocessing pipelines, the ones using those established automated artifact removal methods like ICA that we talked about, the ones designed to give you the purest signal. They don't always help the deep learning models. In fact, sometimes these advanced cleaning steps are actually detrimental. They make the AI perform worse.
Wait, what? You're telling me all that hard work, carefully scrubbing the data, getting rid of the noise, might actually hurt the AI's ability to learn?
Yeah.
That is brilliantly, critically ironic.
It is, isn't it? And the implication is pretty profound. There's this growing suggestion that the EEG artifacts themselves, the eye blinks, the muscle twitches, maybe even other stuff.
The stuff we called noise.
Exactly. That stuff might contain subtle hidden information. Information that these deep neural networks, which are amazing at finding complex, non-obvious patterns, can somehow pick up on and use.
So the noise isn't just noise.
It seems maybe not, at least not to the AI. It's like the artifacts are whispering secrets that contribute to the model's overall performance. Secrets that our human-designed filters, aiming for purity, are just silencing. It really makes you question what clean data even means in this context.
Wow. Okay, so what does this all mean for you, the listener, trying to get your head around these complex brain signals and how scientists study them?
Well, I think it means that understanding EEG isn't just about recording the brain. It's also about critically understanding what isn't the brain, the interference, and then making really informed, sometimes quite counterintuitive choices about how you deal with that interference.
Right. The best approach isn't always the one that seems cleanest on the surface.
Exactly. This deep dive really throws up a fascinating paradox, doesn't it? Our intense search for perfectly clean data might, in these advanced AI contexts, actually lead us to throw out valuable information hidden in the mess.
It definitely challenges our assumptions about data purity versus actual utility, especially as AI gets better at finding patterns we just can't see ourselves.
It's a powerful reminder, I think. Sometimes the perfection we aim for can blind us to unexpected value in what we initially dismissed as just noise.
Which leaves us with a provocative thought, doesn't it? What other noise in our lives, in all the data swirling around us every day, might we be filtering out too quickly? What hidden information might be lurking in the stuff we usually discard, just waiting for something or someone sophisticated enough to actually hear it?